Midterm 2024.
Hi, I'm Hallie.
And hi, I'm Nicole.
Our experiment. We tested how does concentration affect enzyme activity. Background knowledge. The four macromolecules are proteins, carbohydrates, nucleic acids, and lipids. Proteins. Their monomer is amino acids and their polymer is a polypeptide chain. Here's the structure of a monomer and polymer. Their function consists of structural support, transporting molecules, and antibody production. Nucleic acids. Their monomer is nucleotides and their polymer is DNA and RNA. Here's the structure of a nucleic acid monomer and polymer. Their function consists of storing and transmitting genetic material. Carbohydrates monomer is monosaccharides and their polymer is polysaccharides. Here's the structure of a carbohydrate monomer and polymer. Their function is to serve as a primary source of energy and provide glucose for cells. Lipids. Their monomer is fatty acids and glycerol, and their polymer is phospholipids. Here's the structure of their monomer and polymer. Their function is to serve as energy storage, signaling molecules, and more. Enzymes. Enzymes are proteins. Proteins fold in 4 different ways. Here's a diagram of primary, secondary, tertiary, and quaternary structures. Proteins fold in these ways because it is key to their function. Their amino acid sequence determines their structure, and if they don't fold properly, they won't function appropriately. Did you know? Misfolded or unfolded proteins can lead to diseases such as Alzheimer's, Parkinson's, and cystic fibrosis. Our experiment. Our lab consisted of a rattlesnake venom simulation. We tested how does concentration of substrates affect the activity of rattlesnake venom as an enzyme to deactivate its target. Hypothesis. As the concentration of the substrate increases, then enzyme activity increases. The control group is when the enzyme is to substrate ratio is 1:1, so in our case it is 0.2 to 0.2. Our control groups are trials 1 to 2, and here's our data. Our control variables are the amount of enzyme, temperature, water, pH, type of flask, and the same materials used. The independent variable is the substrate concentration, and the dependent variable is the time for reaction. Conclusion. Our data shows that as the substrate concentration increases, the enzyme activity increases, then decreases, and levels off. This is supported by our reaction times.
There are 2 kinds of bacteria, gram-positive and gram-negative. Gram-positive have one phospholipid bilayer and a very thick layer of peptidoglycan. Gram-negative, on the other hand, have a phospholipid bilayer and then a thin layer of peptidoglycan and then another phospholipid bilayer. Antibiotics are a chemical that enters the bacterial cell and damages the enzyme that builds the layer of peptidoglycan. This breaks down the cell wall and causes the cell to explode because of its internal pressure. In antibiotic-resistant bacteria, another enzyme is present that breaks down the antibiotic before it can damage the bacteria that builds the cell wall. Our medical intervention involves the use of radiation therapy to kill bacteria or damage the bacteria that are infecting a patient. In the early stages of a bacterial infection, small doses of radiation can be used to disrupt the DNA of the bacteria. This would help terminate the genes that code for these antibiotic-resistant enzymes and would also disrupt the plasmids that can be transmitted between bacteria because they often carry these genes. Another use of radiation therapy in treating bacterial infections could be to attach gold atoms to monoclonal antibodies. The patient could then take these antibodies, which would attach to the antigens of the bacteria cells. Then, when subjected to radiation, the gold atoms would heat up significantly and the bacterial cells would explode as a result.